All right, so in this video, we're going to look at proteins, and proteins run your body. They are the key molecule to getting your body to do anything it needs to do, and not just your body, but really any organism. And so what are proteins made out of? They're made out of carbon, just like all of our macromolecules, obviously hydrogen, oxygen, nitrogen, and some sulfur, which makes a cool little bond that we'll talk about. So what is the biological purpose of proteins? Really, there's a lot of things. They help with growth and maintenance. They're catalyst for biological reactions. We're going to look at that on this whole side of our paper. They're chemical messengers, so hormones and neurotransmitters. They're how your neurons talk to each other. They're how your cells can talk to another cell far, far away. And they provide structure. Think about your skin is made out of a lot of proteins. Your muscles are obviously proteins, stuff like that. Okay, so their chemical structure is, their basics is they're made out of amino acids. So the monomer of a protein is an amino acid. And so one amino acid has this sometimes like COOH group, or a carboxyl group on one end, a central carbon, and then it has an amino group, hence an amino acid. So it's carbon, carbon, nitrogen, and then these two hydrogens would stick off. One of these oxygens is double bonded, and one's just an OH group. And then off the central carbon is just a hydrogen. And then this is where the difference comes, is we just write it as a general R group, but this varies for every amino acid. And you can look up the amino acid chart in your book. You do not need to know the different amino acids. You just need to know that there's 20 different ones, and they vary because their R group is different. And some are polar, and some are non-polar, and some are acidic, and some are basic, and some are ionic, and some aren't. Some are hydrophobic. So they're all different, those 20 different amino acids. So then the question becomes, how can 20 different amino acids make all the different proteins in the whole wide world? And really think about it this way: how do 26 letters make all the letters in the English language different? It's just the same. It's the same idea. So they're in different arrangements. The 20 amino acids are in different arrangements that are coded for by our DNA. They're in different arrangements or different order, and proteins are different sizes. Some are really, really ginormous, and some are tiny. Same thing with words, right? Some words are tiny, I, a, to, and then some words are ginormous. So same thing here, just different rearrangements and different sizes. So protein structure is really key. And protein structures are held together by these 4 different type structures that we're going to look at here next. So the first one is the primary structure, which is the sequence of amino acids. Okay, and this is held together by covalent bonds. So you're going to covalently link. When you're making a protein in a ribosome, you're going to covalently link amino acid to amino acid to amino acid to amino acid, held together by covalent bonds. It's actually happening between this and this amino group, and then a carboxyl group from another one. Okay. In the secondary structure, you get 2 structures forming. You get what's called an alpha helix, which looks usually represented by like a helix, or a beta pleated sheet, which kind of zigzags. And in our secondary structure, this is interactions along the backbone. Along the backbone. So the backbone is the part right here, this part, not the R group, but so it's interactions. So you can see there's some polar regions and there's some polar regions. And so there's some polarity. And so there's some polar bonding, hydrogen bonding happening along those R groups. And so it's a lot. So the chemical structure, the bonds that are happening is hydrogen bonds. Hydrogen bonds along backbone. And then in the tertiary structure, this is where we start to get into the 3D or the three-dimensional folding. And you start to get what's called a globular protein forming. And this is interactions with side chains. So interactions, I'm actually going to put with R groups. And this can be all the bonds. So we're going to have H bonds, hydrogen bonds. We can have, I said some of those, our groups are ionic, so we can have ionic bonds form. We can have hydrophobic interactions. And then we can also have these ones, I'm going to just write it right here, but it's really up here. So they're disulfide bridges, and they occur between two sulfur R groups that have sulfur in them, and they form, it's a type of covalent bond, so it's a pretty strong bond. And then in the quaternary structure is you get more than one amino acid chain bonding. And it's the same as tertiary structures, the type of bond. A good example of this is tertiary structures. Hemoglobin, which carries our oxygen, so it's found in red blood cells, it carries our oxygen. It is 4 different amino acid sequences, and so 4 different secondary structures and tertiary structures, and then they all come together to form one quaternary structure that makes one protein that does a job in our cells. And then when we look at, so what are some different types of proteins? So some are called structural proteins, and they form cells or bodies. And so some functions of this is they form the basic structure. You're going to see proteins in cells making your cytoskeleton. There's proteins in membranes that help with transport. They provide protection, so things like skin and stuff like that. And they make up the appearance, things like muscles and all that kind of stuff is the idea of our structural proteins. They hold things in place. They move. They're able to help things move and all that kind of stuff. And then the other one that we're going to look at is our friends, the enzymes. We're going to spend a lot of time talking about enzymes and and they're a type of functional protein. And what they are going to do is they make your body work. Okay. So these are our work guys, and they're going to catalyze reactions. And so we have what's called the lock and key mechanism. So if you're thinking, this is a really bad picture that I'm about to draw, but I will direct you to one in your Edpuzzle of where to go. But this is a protein, okay? So this is my enzyme here, enzyme. And I'm going to have some substrates come in to these active sites, and it's a lock and key mechanism. These substrates are very specific to this specific enzyme. So I'm just going to say that these are 2 sugars. And what I'm going to do is they're going to force, it's called the induced fit model. They're going to force those 2 substrates together in such a way that they're going to have to chemically link. And so then what's going to end up happening is my enzyme, which is, because it's a catalyst, it won't be used in my reaction. So it will be available again, will form, and then I will form, here's my 2 substrates that are now chemically linked together. And this enzyme can go and do that again and make a new molecule and make a new molecule. This could be a disaccharide, 2 sucrose molecules linked together. 2 glucose molecules linked together. Or it could be a lipid getting linked together, or it could be 2 nucleotides getting linked together to form a DNA. Any of those things could happen. Okay. It can also, an enzyme can also break bonds. So it can force them in that induced fit model. If that's the type of enzyme that it is, it can break a bond. Okay. And so how does it do that? How does the enzyme catalyze that chemical reaction? It's really, we're just going to summarize what I've kind of said, is the substrate bonds to the active site. And it's obviously not a very permanent bond. It's a very temporary bond because this enzyme is going to be used again and the substrate's going to go. And the activation energy, which we'll look at in a second, required is greatly reduced. Okay. So let's look at what an activation energy is. So if I make a graph of a reaction, and so this is the energy of the reaction, the energy needed for a reaction to occur, and this is the pathway, or really the time. Kind of made that really low. And so over here, we're going to make it like this. So here's my reactants in my equation, and here's my products. And I have to, in order to make this reaction to occur, I have to get over this hump. I have to put in a little bit of energy in order to eventually release energy from this reaction in this case, because this is an exothermic reaction. And so this is the normal kind of reaction and activation energy. And what it does, what an enzyme does, is going to be along this dotted line. It lowers the activation energy. So it lowers that hump. So I'm going to put with enzyme, and then this line up here is without an enzyme. So that's all an enzyme does, but it's key. Without enzymes, we wouldn't be able to do many things. And enzymes, sorry, back up here. Enzymes always end in ASE. Not always, I shouldn't say that, but generally speaking, they end in ASE. So if you see a molecule that ends in ASE, you kind of know what it does. So sucrase is an enzyme that breaks down the sugar sucrose. Lactase is the enzyme that breaks down the molecule lactose. So if you're lactose intolerant, generally you don't make enough lactase in your body. There's all kinds of other ones. We're going to learn about DNA polymerase. We're going to learn, there's so many that we will learn about. The problem is that enzymes are sensitive, and really, actually, generally speaking, proteins in general are sensitive, but enzymes are the ones that we need to do this work in our cells. And so they can denature, or another word for that is the destruction of secondary and tertiary. I'm totally abbreviating those. Tertiary structures. They break apart. When we denature, it breaks apart all those hydrogen bonds and hydrophobic interactions and ionic bonds. It just breaks it all apart. And so if the enzyme no longer has its shape, it no longer has its structure, it can no longer function. Okay, so no structure equals no function for this enzyme. That structure is key for it to do its job. Just like up here when we talked about the lock and key mechanism, it was key. It was very specific, and if it changes its shape, it changes its job. It can no longer do that same job. Okay, so what are things that can affect it? One is temperature. Really specifically, too hot. An enzyme has an optimal temperature that it will work at, and if it gets too hot, those molecules are colliding more and more, and those hydrogen bonds and things will get broken apart. And so we've seen that, like when if you've ever get heat stroke, or what's happening is your enzymes are denaturing. Another one is a change in pH. Obviously, if you're changing pH, you're changing the amount of hydrogen in your liquid. So too high or too low in this case. Too high, too low. You're changing the amount of hydrogen, things like that. So a lot of, there's all kinds of examples of too high and too low pHs in like an ecosystem, an aquatic ecosystem, if you change it, in a soil ecosystem, things like that. Another one can be poisons, because poisons can act like inhibitors, and we'll get into in a different area in class. We'll look at different types of inhibitors that can compete for binding sites. They can bind onto that enzyme, and then one specific example of poison is salt. Can change can denature an enzyme as well, or we can get inhibitors, things that block the binding site, block the active site. Salt will just denature because you're adding a bunch of ions to the system. But it could be a type of poison. All right, so that's a lot, and we're going to do some labs and lots of practice in class to make sure we understand it.